Those of you who don't know me, my name is Pollnac, and I'm a postdoc in Rich Smith's lab. And today I'm going to be talking to you a little bit about some of the research that we've been doing on the effects of genotypic diversity on forage stand yields.
Love it.
I can't.
Oh.
Get louder. Sorry, I'm dealing with losing my voice, so I'm sorry if you can't hear me in the back. I'll do my best. But in this work, actually, Andre alluded to a little bit earlier when he was talking about research and part of the project that he's doing looking at grazing season extension. So there were two pressing issues that really motivated the initiation of this project. And the first was a needs assessment of organic dairy farmers in the Northeast, where they indicated that they're really interested in research that was aimed at extending the growing season, so that they could ensure that they would comply with the new National Organic Program pasture rules. Second of which is the potential for increasing variability in temperature and precipitation with climate change, which presents additional challenges for maintaining high quality and productive pastures from year to year with that anticipated additional variability in climate. And so there's one tool that we're looking at now that we think can sort of deal with both of these issues, and that's the addition of genotypic diversity to forage crops. So to define what genotypic diversity is, a genotype is just another name for a cultivar. So with something like perennial ryegrass, we've got the cultivar Remington, cultivar and genotype are synonymous. So when we're talking about genotypic diversity, we're really just talking about cultivar diversity. And in practice, that just means growing several cultivars in the same field at the same time. The theory behind using genotypic diversity to perhaps lengthen the growing season or increase productivity lies in the fact that individual cultivars have these ranges of environmental tolerances. It's displayed with this little conceptual diagram here. We've got production on our y-axis and environmental conditions on the x-axis. So an individual cultivar during the growing season, when conditions are favorable for growth, has a range of conditions in which it functions optimally, so it produces the maximum. And then in other parts of the growing season, when it's a little cooler and drier, what have you, it doesn't produce at its optimum. So you've got this change in production throughout the growing season based on the environmental tolerances of that cultivar. And if you expand that to a longer time scale, so now we're looking at multiple years here and the duration of growth or the length of grazing season. If you have a year where the average conditions during the growing season are in this really favorable range, you're going to have a good production year. But if you have another year where those conditions are in a suboptimal range, that results to having a poor production year. So that over time, because you've got this one cultivar with this set number of environmental conditions or a set range of environmental conditions that it likes, as that climate varies from year to year, your production is going to vary from year to year as well. And so now introducing that concept of genotypic diversity, so now instead of just growing one cultivar in our field, we're going to grow three. And the key here to make this useful, we believe, is that you need to include cultivars that have slightly different environmental tolerances. And the first thing that you might notice that this does, is it takes this length of the growing season here and extends it slightly, because you've now got two additional cultivars with slightly different environmental tolerances that can tolerate those conditions at either end of the growing season. It also lengthens the amount of time during that growing season where production is closer to peak, so that gives you an overall increase in production over that one growing season. So extending this genotypically diverse forage crop on a longer time scale, again, you can see how that might increase the stability of yield from year to year, because by including these additional cultivars with different environmental tolerances, you're essentially providing yourself with some insurance that regardless of how the climate fluctuates, you're probably going to have something in your forage crop that can deal with those environmental conditions and produce at a higher level. So that's one way of dealing with environmental uncertainty over time as well. The studies that we're doing at UNH, we're working with perennial ryegrass, but this concept here should be transferable to other forage crops as well. And so the short-term benefits of genotypic diversity, we believe, are increased forage productivity throughout the growing season, The long-term benefits of genotypic diversity would be that increase in yield stability from year to year. So we established some experimental pastures at UNH where we planted monocultures and mixtures of perennial ryegrass. We did this at 2 locations. One was the UNH Kingman Farm, and that study's been going for 2 years. The other is at the UNH Organic Dairy Research Farm, and that study's been going for just 1 year. And that Organic Dairy Research Farm study, which Andre alluded to earlier, is also repeated in 3 other states, Vermont, Maine, and Pennsylvania. The measurements that we took were pretty straightforward. So throughout the growing season, the pasture would be measured for grass height on a weekly basis, and once the rye reached a certain height, biomass would be sampled from all of our experimental plots. That biomass sample would be brought back to the lab. Grass would be separated out from weeds. The sample would be dried and weighed. And once those samples were harvested from the field, the entire field was mowed and baled to simulate grazing by cattle without the additional unpredictability of having cows running around out there. And this was done several times throughout the grazing season as well, or growing season in this case, since we didn't have real grazing. So throughout the summer, we went out and harvested 5 times from this particular field. The 3 measurements of forage stand productivity that we were looking at were dry matter, yield index, which is the complement to dry matter, which I'll sort of explain later, and the response to cutting, or the growth rate after cutting. Getting into some of our preliminary results, this is from the Kingman Farm study. We've got a plot here of ryegrass dry matter production versus cultivar diversity. So we had mixtures of 3 and 6 cultivars along with monocultures. And you can see here that after our statistical analysis, we did find that there was a statistically significant difference between the mixtures and the monocultures, where the mixtures were producing significantly less dry matter throughout the growing season than either of those mixtures with 3 or 6 cultivars, which supports our hypothesis that mixtures would be more productive throughout the growing season. Looking now at data from the Kingman Farm study, or the Organic Dairy Research Farm study, we've got, again, total dry matter on our y-axis, but we've now got treatments along the x-axis, and I'll explain these a little bit. We started with a very simple treatment, which was just a monoculture of the recommended cultivar. Then in all of the other treatments, except for treatment 6, which was a commercially available mix, we maintained that recommended cultivar in the mix, but just added additional cultivars to tweak things like the heading date range or the winter hardiness range of the species that were planted out in the field. And these numbers in parentheses are just the numbers of cultivars within each one of those mixtures. And so, for example, going from treatment 1 to treatment 2, this is the monoculture. Treatment 2 had that recommended cultivar, but it added 2 additional cultivars, one with an earlier heading date and one with a later heading date. So the statistical analysis of this wasn't quite as conclusive. We've got a p-value here of 0.52, which indicates that there's no significant difference between the dry matter yield of any of these different treatments. But on the positive side, at least that meant that mixtures weren't any less productive than the recommended cultivar. And just looking at the averages, a lot of them actually appeared to have slightly higher production, even though it wasn't statistically significant. The interesting thing that we noticed, now we're just looking at dry matter production in May, so the first harvest, early on in the growing season. You can see our p-value from that analysis went down to 0.15, which is closer to significance. So we're seeing that differences in dry matter production between the monoculture and these sort of strategically designed mixtures might be more apparent earlier on in the growing season, or more likely to manifest earlier in the growing season, which gives us some hope that these strategic mixtures that have been designed might actually work towards that goal of extending the grazing season, at least at the early end. Now, to complement that dry matter data, we looked at something called the yield index. I'll just briefly explain what that is. So suppose we're going to make a mixture with 3 cultivars. First thing that we might do is look at how those cultivars yield in monoculture. So for this example, we'll assume that we have these 3 cultivars. And we know that when they're grown at a certain seeding rate in monoculture, they yield about 300 grams of dry matter per meter squared. Now take those 3 cultivars, mix them together in equal parts in the seed mix, and plant them as a mixture. And we can predict that because we've got equal parts of those 3 cultivars in the mix, if we plant it at the same seeding rate, we're also going to get a yield of 300 grams per meter squared, all else being constant. In reality, we could observe a couple of other things happening. We might see that our mixture actually yields more than we expect, or it might yield less than we expect. And so what a yield index does is it just gives us a simple way to characterize that. So to get a yield index, we just take our observed yield and divide it by the predicted yield. If we get a value of 1, that just means that the observed is what we expected it to be. It's the predicted yield. If we get a value greater than 1, that indicates some sort of over-yielding going on. And if we get a value of less than 1, it indicates some under-yielding going on. So we're getting less than we would expect, perhaps due to competition between the different cultivars. At the Kingman Farm study, we did see significant over-yielding, which is really interesting because this indicates that those increases in dry mass that we saw weren't just because, by chance, we were including more productive cultivars in the mix. This indicates that we're actually getting a response that is greater than the product of the yields of all those cultivars in the mixture. So we're seeing something additional going on here besides just throwing a really high-yielding cultivar into the mix. And that could be due to some sort of facilitation between cultivars. They have a Y with a question mark there because we're not really sure why that's happening at this point in time. It's something we need a little bit more time to figure out. The last result that I'm going to touch on here is our response to cutting. And so this graph is just the fall growth rate expressed in centimeters per day versus treatment. I'm just looking at the fall growth rate here because we didn't actually start measuring this until the fall. In subsequent growing seasons, we're going to be measuring this all throughout the grazing season. We've got those same 7 treatments on our x-axis. And here what's really encouraging for us is we see a statistically significant difference between the recommended cultivar and this treatment 3, which has the greatest range in heading dates. And so this treatment here is yielding or has a growth rate that's significantly higher than the recommended cultivar, which indicates that it has the potential to be a more productive late season pasture mix and maybe even extend the grazing season a little bit more into the fall, which is an encouraging result. So in summary, we saw that increasing the number of cultivars increased dry matter production. Purposefully designed mixtures weren't necessarily less productive than the recommended cultivar monocultures, but this is just the first year of that study, so treat those results with a little bit of skepticism, perhaps. Mixtures over-yielded, but we need more time if we want to figure out exactly why that's happening. And lastly, we did see that a purposefully designed mixture had a better growth response than the recommended cultivar late in the growing season, which could, again, allow us to have a little bit of grazing season extension. We do think that genotypic diversity shows promise as a way to increase productivity and extend the grazing season. And please stay tuned for more information on yield stability over time. Since these projects have been going on for a relatively short term, we can't really say anything about that year-to-year stability at this point, but hopefully we'll be able to speak to that in the future. And again, this study used perennial ryegrass, but the ideas should be transferable to other forage crops that people might be working with as well. I'd like to thank all of our field assistants and funding agencies, and if there's any time for questions, you can just catch me.